Welcome to the third video on the unit on microbial control. In this video, we're actually going to talk about different classes of antibiotics, and we're also going to compare them to some antivirals, antifungals, and antiparasitics. Remember, you don't need to memorize what every single antibiotic does. I more want you to understand the different classes and what their general targets are so that you can understand the difference between different types of antibiotics. Let's go ahead and get started. Remember, antibiotics are selectively toxic because they target certain reactions or structures in bacterial cells that are not found in human cells. So the targets of antibiotics are important for their function. The two most common targets are the cell wall, the peptidoglycan, and the ribosomes. Remember the 70S ribosome compared to the 80S ribosome. So common cell wall disrupting antibiotics are the beta-lactam antibiotics, such as things like penicillin or amoxicillin, anything that ends in -illin, while drugs such as tetracycline or azithromycin, your Z-Paks, function to prevent translation by targeting the ribosome. Other areas that are targeted by antibiotics include nucleic acid synthesis. This one has less targets because most of the enzymes in DNA replication are homologs between prokaryotes and eukaryotes, except for one key enzyme, DNA gyrase, that was involved with the supercoils of bacterial DNA, doesn't exist in eukaryotic cells. And so DNA gyrase tends to be a common target for things like ciprofloxacin. Other targets are metabolic pathways. There are a handful of metabolic pathways in bacteria, things like folic acid synthesis, that the enzymes in the pathway are different than human enzymes. And so those metabolic pathways can be targets of drugs. And then the last is the plasma membrane. These are a little bit less selectively toxic, so they're typically used more as topical antibiotics, more a little bit like antiseptics, mainly because your cells also have a plasma membrane, so things that disrupt the plasma membrane could also harm eukaryotic cells. We're going to take a look at each one of these major target areas in a little bit more detail. The peptidoglycan of the bacterial cell wall is a common target for many antibiotics. This is because without a cell wall, bacteria cells will lyse due to osmotic pressure. If they can't form the proper peptidoglycan, those antibiotics will be bactericidal and they will lyse due to osmotic pressure. However, the cells do have to be actively growing in order for these to function because they prevent the formation of peptidoglycan. They don't destroy peptidoglycan that is already there. The two common classifications of antibiotics that are cell wall inhibitors are the beta-lactam antibiotics, such as things like penicillin, methicillin, ampicillin, and the glycopeptides, the most common of which are vancomycin. You'll notice that both these classes of antibiotics are preventing this step here, where we have a molecule, a building block of peptidoglycan. So we've got our NAM and our NAG sugars, and then we have our pentapeptide that's supposed to get cross-linked to the rest of the peptidoglycan layer. Both beta-lactams and glycopeptides prevent this cross-linking from happening, so we can't build that new peptidoglycan wall. However, the beta-lactams do it by inhibiting the enzyme that catalyzes this reaction, whereas glycoproteins interfere by physically binding to the peptidoglycan, preventing it from being able to attach to that growing peptidoglycan cell wall. So we're going to look at the beta-lactam antibiotics in a little bit more detail. These antibiotics function by inhibiting an enzyme called transpeptidase, which is also known as penicillin binding protein because that's how it was actually discovered, is it's the enzyme that penicillin binds to and how penicillin functions. So we can see here in the image, this is the NAM-NAG subunit of peptidoglycan with the pentapeptide amino acids. And this is the enzyme, the transpeptidase, that's going to catalyze the formation of the cross-linkage here between this set of peptidoglycan and this set of peptidoglycan. So we can see that linkage that's now formed. The way beta-lactam antibiotics work is they bind to the enzyme directly, and once they're bound, that enzyme can no longer function. And so without the enzyme, we can't form the cross-links, which means we can't attach one chain of peptidoglycan to another chain of peptidoglycan and make the full cell wall. When you look at beta-lactam antibiotics, They all have a structure here that's this four-sided ring with a nitrogen. That's the actual beta-lactam ring, and it's that structure that makes these beta-lactam antibiotics. The penicillins are the natural antibiotics, and those were the ones that were discovered from Penicillium notatum. And we've since then turned some into semi-synthetic antibiotics, things like ampicillin and amoxicillin that you might have heard of. We then have other classes of beta-lactam antibiotics that also have a beta-lactam ring, but have a more broad spectrum for their activity. And those include the cephalosporins, which we're now up to a fifth generation, so we've continued to chemically modify these, and the carbapenems. The carbapenems are the newer antibiotics. A lot of these just came in the market over the past 10 years, and they're typically used and reserved for things that are antibiotic resistant. And so if you have microbes and they're causing infections and they're resistant to other types of antibiotics, we'll use the carbapenems as a last resort. However, we're already starting to see things like CRE, which stands for carbapenem-resistant E. coli, and so we're already starting to see resistance develop to these newer classes of beta-lactam antibiotics. Both your cephalosporins and your carbapenems will work against both gram-positives and gram-negative organisms, whereas your penicillins are more traditionally used against just gram-positive. Another broad target for antibiotics are the inhibitors of protein synthesis. So overall, these antibiotics are a little bit more broad spec than those that inhibit cell walls, so they cover a little bit more of a broader range of gram-positive and gram-negative organisms. And the majority of them are actually bacteriostatic. And so if you stop protein synthesis, oftentimes the bacteria can't grow anymore because they can't make their enzymes and make the proteins that they need, but they already have some functioning proteins, and so they're able to just kind of hang out. They can't grow and divide, but they're also not dying, which makes most of these bacteria static. They're also selectively toxic because remember there's a bit of a structural difference between the prokaryotic 70S ribosome and the eukaryotic 80S ribosome. If we look at some of the classes directly, you can see the first class listed there are the macrolides. These actually stop protein synthesis by interacting with the 50S subunit, so the large subunit of the ribosome. A good example of macrolides are azithromycin, which makes up your Z-Pak antibiotics. Macrolides are a little bit different because they have a very long half-life, so they remain in the tissues for a long time. This is why Z-Paks can exist. As Z-Pak, you typically take maybe 3 or 5 antibiotics, so once a day for a short period of time. That works because the half-life is so long that the antibiotic stays above the MIC in the tissues themselves. If you look at other inhibitors of protein synthesis, there are some that then bind to the 30S, the small ribosomal subunit, instead of the large ribosomal subunit. These are things like aminoglycosides, which are things like streptomycin or gentamicin. These are going to actually result in protein still forming, but they're not able to really be proofread. They get in the way of how the protein functions and how the tRNAs come in, and so they're really faulty proteins that can't actually function. And then the last class are the tetracyclines. These are also binding to the small ribosomal subunit, and they completely inhibit protein synthesis because they stop the tRNAs from coming into the ribosome at all. If the tRNA can't bring in the antibiotic, no proteins can be made. An example of tetracyclines are tetracycline and also doxycycline, so things that end in that cycling. Keep in mind that most of these, other than the macrolides, are bacteriostatic antibiotics that aren't actually killing the cells themselves. Mentioned in the overview slide that there aren't very many classes that inhibit nucleic acid synthesis because most of the DNA replication or transcription proteins are similar between prokaryotes and eukaryotes. So there's a few unique targets. The quinolones are targeting that DNA gyrase, remember that I mentioned, that's involved in supercoiling, and those are things like ciprofloxacin. There's also antibiotics, mainly the metronidazoles. It has the brand name of Flagyl, that actually nicks DNA. So they damage the DNA directly. But these are interesting, and the reason they're selectively toxic is that Flagyl is actually a prodrug. So the compound itself is non-functional. It's not till it gets into a cell and gets metabolized that it becomes the functional drug that then breaks down the DNA. The key with Flagell is the metabolism that allows it to become activated only occurs in bacteria that are anaerobic or microaerophilic. And since your cells are aerobic, you can actually use this drug and it doesn't damage your DNA, but it will damage the DNA of anaerobes. You should also notice that means that this has a relatively narrow spectrum. It's not going to work against any of your aerobic bacteria, only your anaerobic ones. The last target we mentioned was the plasma membrane. So remember, the plasma membrane has much lower selective toxicity, and so these tend to be used relatively sparingly. The main example are the polymyxins. These are things that are found in stuff like Neosporin. They're also used often in bandages for burn victims, so the antibiotic is embedded in the bandage itself to help prevent wound infections. So again, mainly as topical antibiotics so that they're not getting into the body and possibly harming your own plasma membranes also. Interestingly, the last one on the list, the platysmycin, this is I've put on here not because you need to know how it functions, but to highlight that this was one of the very first antibiotic classes discovered in the past 50 years. And so when we talk about antibiotic resistance, we're going to talk about the fact that there really haven't been any new antibiotics. All the antibiotics I've been talking about in this video were really discovered back in the 40s, 50s, and 60s. And so the idea that we finally have at least one new antibiotic class, especially one that can work against Mycobacterium tuberculosis, was a big deal when Merck first identified this back in 2006. Now we're going to talk about some of the other antimicrobial agents, but we're really going to go over a general overview of how these antimicrobial agents may function. And I really want to focus on how they have differences in chemotherapeutic indices and selective toxicity compared to the antibiotics. So when you look at antiviral agents, we have many fewer antiviral agents, so there is a lot less ways to combat viral infections with drugs than there are bacterial infections. And this is mainly because the viruses use your own cell's mechanisms for replication. So there's not a whole lot of viral specific targets, which is what gives us that selective toxicity. Instead, there's a couple key parts that mainly antivirals try to target. So oftentimes, they will try to prevent attachment. Remember, you have that unique interaction between the spike protein of the virus and the receptor on the cell. If you can prevent attachment with a drug, you can prevent viral replication. There's often many drugs that will try to prevent viral release. And so as that virus tries to bud out of the cell, it prevents the release of the budding virus. And if they can't get out of the cell, they can't go and infect other new cells. Within the cell itself, that really depends on the virus. So for example, if you have a retrovirus, which has a reverse transcriptase enzyme, our cells don't have one of those. And so many of the HIV drugs will target the reverse transcriptase of HIV because that can provide some selective toxicity since it's not found in our cells. The same thing can happen for RNA viruses where you can sometimes target the DRP, that RNA-dependent RNA polymerase, that again was unique to viral cells. This is also why most antiviral agents are specific to the virus itself. So for example, you have drugs that work against influenza. You have other antivirals that work against HIV. But you don't have a broad spectrum antiviral that works against lots of different viral agents because these targets are much more specific to the virus itself. When we look at the fungi and the parasitic pathogens, those are eukaryotic cells. And so these eukaryotic pathogens have very few fundamental differences compared to our human cells because we're both eukaryotes. What that means is it's a lot harder to get that selective toxicity, to find drugs that would be targeting just the fungal or parasitic pathogen and not harming our human cells. For the fungi, there are a couple of targets we can try to take advantage of. Remember, all fungi have a cell wall, and that cell wall is made up of chitin and then beta-glucan sugar layers. These structures aren't found in human cells, and so those can be a possible target for antifungal agents. The other key target is the ergosterol, which is found in the membranes of fungal cells. You can see the structure of ergosterol here and how it compares to that of our cholesterol. There's clearly differences there, which means there's differences in how the molecule itself is made and what it looks like. Those differences can be targets for antifungal agents. Because of these two main structures, there are a couple of broad-spectrum antifungal agents that will target multiple fungi species and do not harm our cells. However, for your antiprotozoal or antiparasitic agents, those protists that we've talked about, the helminths that we talked about, there are no broad-spectrum agents. What that means is if you're trying to make an antiparasitic agent, it's typically going to be targeted to something that's specific about that parasitic pathogen and isn't found in other parasites. For example, we have anti-malarial drugs that will target the malarial parasite, but don't target other parasites. And this is because there really are no differences that are found in all parasitic cells, but are not found in human cells. And without those differences, you can't develop a broad-spectrum drug.